So for the next talk, we're going to move from the species level to the population level with a talk by Parul Johri from Indiana University.
Hi, I would like to start by thanking the organizers for this opportunity. So today I will be talking about population genomics of Paramecium species. Quantifying within species variation is important for a population level understanding of key processes in evolution. In the last decade, there have been a burst of population genomic studies that have allowed us to understand factors that shape patterns of variation, like recombination, mutation, drift, and selection. More specifically, population genomics has allowed us to detect spatial structure and local adaptation in populations, as well as recent species-specific positive selection and estimating the strength of selection operating on various classes of sites. Most such studies, however, have been restricted to multicellular organisms. Although unicellular organisms span all eukaryotic subgroups and comprise most of the phylogenetic diversity, as can be seen in this eukaryotic tree of life, they are heavily understudied. Genome-wide measurements of genetic variation in unicellular species have been largely restricted to either parasitic or free-living organisms that belong to mostly the fungi or the plant clades. So such dearth of analysis in the unicellular species prompted us to assess genetic variation in 5 different species belonging to the Paramecium genus that belongs to the alveolates, and that is quite distant from the other well-studied clades. So specifically, we focused on the Paramecium aurelia complex, which you just heard about. This is, again, a set of 15 morphologically identical, closely related species. And 2 of the unique genome features of the Paramecium complex is that they're highly streamlined or compact, and that they have about 40,000 protein coding genes as a result of 2 successive whole genome duplications. And most of the aurelias retain still about 50% of duplicates that were created by the most recent whole genome duplication. So now Paramecium, as you saw, is also an upcoming model organism to study the evolution of duplicates. So we decided to assess genetic variation in the macronucleus, which is the transcriptionally active nucleus in Paramecium, for 5 different species. So 3 of the aurelia species, and 2 of the closely related outgroup species, P. multimicronucleatum and cauditum. So we collected about 10 to 13 isolates of all these 5 species. They were sampled worldwide, and all 5 species are cosmopolitan. So we found that all 5 species were monophyletic, as you can see, with biaurelia, tetraurelia, sexaurelia, cauditum, and multimicronucleatum. The 2 outgroup species we observed branched off into deeper, clearer, deeper clades, which might have some cryptic species variation, but these would have to be confirmed with further mating tests. In addition, all the starred isolates mapped to less than 75% of the reference genomes. So we conducted all of our analyses with all of the isolates, as well as a conservative subset called the P. cauditum subset and the P. multimicronucleatum subset for all of the analyses. And we find that the results consistently show the same trends when done for full sets and subsets. So interestingly, in all the 5 species, we found a lack of global population structure. Here is a plot of the geographical distance versus the pairwise divergence of isolates in P. sexaurelia, and we see no correlation. Similarly, we see the same thing for all the 5 species, and these were also confirmed by PCA plots. The Synonymous cite nucleotide diversity, which is a measurement of genetic variation. was, among the aurelias, it seemed like the synonymous site diversity was very similar to the other known unicellular eukaryotes. But in the outgroup species, P. chordatum, P. multimicronucleatum, the synonymous site nucleotide diversity is among the highest reported. So for this talk, I'm going to talk about spatial patterns of nucleotide diversity that we evaluated across non-coding regions in the genome. And we also performed gene-based analysis of patterns of synonymous and non-synonymous diversity. So the macronuclear genomes of a Paramecium species are highly compact. So for instance, in the aurelias, the median intergenic length is about 161 base pairs, and the median intron length is 25 base pairs. In contrast, in the outgroup species, in the outgroup species, the median intron length is about 22 base pairs, but the median intergenic length is as short as 41 base pairs. This is interesting because for the purpose of this study, intergenic length is defined as the non-coding region between the start and stop of neighboring genes. So in order to identify specific sites under stronger evolutionary constraints, we evaluated the nucleotide diversity across the non-coding regions, starting with the introns. So for the introns, we split each of the introns in midway and then align them by position based on the 5' and the 3' splice sites. We then averaged the nucleotide diversity for each of these positions across all the introns in the genome. In this figure, the y-axis is the nucleotide diversity per site. The x-axis shows you the position of the intron, with -1 representing the 5' splice site and the +1 indicating the 3' splice site. The black points show you belong to the introns, and all the colorful points belong to the protein coding regions, with red being 4-fold, blue being 2-fold, and green being 0-fold degenerate sites. So we observed that at least 3 sites from the 5' end and 2 sites from the 3' end that must contribute to the splicing signals are more conserved than the 0-fold degenerate sites on average. We also observed that about 10 base pairs of the protein coding sequence flanking the splice sites are relatively more conserved. This suggests that part of the splicing signal might reside in the coding sequence. And we see the same trend for all 5 species. We next evaluated the same thing for the intergenic regions. So we partitioned all the intergenic regions midway and then aligned them by position based on stop and start codons. In this case, the black position points belong to the intergenic regions, and the -1 represents the stop codon, and the +1 indicates the start codon. So we observed that the intergenic region is constrained up to about 125 to 150 base pairs, suggesting the presence of UTRs or gene regulatory elements in that region. We also observed that there are constraints on silent sites up to 70 to 100 base pairs on both the 5' and 3' end of coding sequences. This can either be because of sweeps in nearby regions, like intergenic regions. It could also be because the 5', 3' ends of coding sequences are under strong purifying selection because of their important role in translation of proteins. A third possibility could be that because these genomes are so compact, the regulatory elements from intergenic regions might actually extend into the coding sequences. Interestingly, the 5' UTR regions that have the putative 5' UTR regions identified in P. tetraurelia are about 21 base pairs in length. So if we assume that in P. chordatum, we also have similar length of 5' UTRs, then we get that all promoter and gene regulatory elements must reside in about 20 base pairs upstream of genes. So on one hand, this definitely suggests that regulatory elements must extend into coding regions. On the other hand, it also raises an interesting question about what is the minimum number of bases required to encode regulatory information about transcription of genes in eukaryotes. So a recent study in yeast has identified a minimal promoter, which is only 116 base pairs long. But this suggests that we might be able to find a much smaller promoter region needed to drive expression patterns in P. caudatum. In fact, most of the P. caudatum intergenic sites must be functional and must be under stronger selective constraints. And we see exactly that when we evaluate these patterns of nucleotide diversity across intergenic regions in all species separately. So we see that in all the aurelia species, the diversity in the intergenic regions stabilizes up to that of the fourfold degenerate sites. But that in the outgroup species, P. caudatum and P. multimicronucleatum, it only reaches that of the twofold degenerate sites. In fact, when we also plot the nucleotide diversity in coding regions by the intergenic regions, we clearly see that in the outgroup species, the diversity in intergenic regions is much lower than that in coding regions. And we know of no other species that has such low diversity in the intergenic relative to coding. Other unicellular species that also have fairly compact genomes, like S. cerevisiae and S. pombe, in fact, have similar ratios as that of the aurelias. So next, we wanted to look at gene-based analysis of patterns of synonymous and non-synonymous diversity. All right, and so our driving questions were whether the selective constraints are similar in paralogs or gene duplicates created by whole genome duplication that occurred 320 million years ago, and whether these selective constraints are similar across orthologs that between deeply diverging species. So we used the statistic pi n by pi s, which is diversity at non-synonymous by synonymous sites, as a statistic for recent purifying selection. So the x-axis here has pi n by pi s for paralog 1, and the y-axis has it for paralog 2. And we can see that for all the aurelia species, these are highly correlated. This overall suggests that the duplicates in the aurelias are not undergoing substantive change in function or even loss currently. We wanted to similarly evaluate this for the orthologs, which are in this case will be two sets, the red and the blue set, because of the whole genome duplication. And we find that in both the red and the blue sets of orthologs, pi n by pi s is correlated between these deeply diverging species. This seems to suggest a low turnover of protein expression, function, or structure, or a number of interacting partners. And we know that this is not a consequence of selection on silent sites or just base composition, because pi s is not necessarily correlated between paralogs and orthologs across all these 5 different species. So anyway, in conclusion, there's a lack of global population structure in Paramecium. This seems to suggest it seems to support the everything is everywhere hypothesis, which suggests that microbes are ubiquitously present everywhere and do not experience geographical barriers. We also observe strong conservation of intergenic regions and conservation of selective pressures acting on paralogs within and orthologs across deeply diverging species. With this, I would like to thank my advisor, Michael Lynch, my collaborators who helped with the project. A special thanks to Lydia and Jeff, and my graduate committee and NSF for funding. Thank you.
I have a quick question. So you removed the weird outliers from caudatum and multimicronucleatum, and yet you have a very high silent-site diversity for this species. Do you think it could be that despite this correction, you still have some cryptic species that are present there?
Well, maybe it's possible, but I don't know. Yeah, there's a possibility, but it's also possible that that's not true because these are cosmopolitan and could have a pretty high effective size. So the synonymous site diversity can be quite high, especially at this global level.